So I've been asked just to take a step back from, we've heard a lot about the importance of quality control, standard procedures, getting the ethical provenance correct. But of course, all of that has to, in order to operate a professional and reliable service, you need to operate within some kind of framework that identifies what the standards are you're going to use and how you're going to deliver them, how you deal with customers, etc. So it covers the whole thing. So I'm going to talk about quality systems, but particularly quality assurance, and the key document in all of this, which is, to me, the quality manual, which describes those standards and outlines the procedures that are going to be used and how you'll assess the results. So firstly, I want to say a couple of other words about quality. It's not necessarily going to be, can you make it the very best it can be? Because that might not be needed. You need to make something that's fit for purpose, and for different applications, that's going to be described by a different set of protocols and tests and characterization. And also, like beauty, quality is in the eye of the beholder. It depends what perspective you're taking. And very often from the point of view of EBiSC, obviously we're taking the user perspective, which is often focused on speed of supply and cost. Is it fast? Is it cheap? Can I get my cells next week? Can I only pay a couple of quid for them or a couple of euros for them? And of course that's not practical always, as we've heard with the difficulties and the cost of running these things. And the scientific quality, surprisingly, is sometimes the last thing that people want. They've seen the name of the cell line, they've seen the description in the catalogue, and they just want it. And even in reviews over several decades, in looking at amongst the European culture collections, when they ran questionnaires, customers were asked if they wanted to have a quality standard established for their cells, if they wanted quality issues to be addressed, and surprising numbers said no. We just want the cells. Give us the cells. I've got to get my paper out. I need to do it quick. Don't mess around. Forget the quality. And of course, they were thinking of the paperwork, not necessarily the scientific testing that's necessary. And also, sometimes customers don't, in certain circumstances, particularly younger researchers, don't fully understand the implications of not having certain tests done for their own work, for their patent applications, for exploitation going down the road. And unfortunately also, as probably a lot of us know here, obtaining cells from a friend down the corridor has led to widespread incidents of switched cell lines, cross-contaminated cells, mycoplasma contamination being transmitted between labs, and as a result, hundreds of thousands, if not millions, of wasted research dollars and euros over the years. And it really is important to do this, as other speakers have explained, the importance of having centralized facilities, doing consistent quality control work. So EBiSC, as you've heard, is aimed to deliver qualified cells for disease studies. That's its customer focus, really, if you like, in terms of establishing the quality. So there's traceability on ethical procurement, good standards, high standards of quality control, and using good cell culture practice, so that we can guarantee reproducible and reliable cell supply, and achieving this under a quality management system linked to industry standards. Because industry is going to be the recipients of these cells ultimately in terms of the testing work that's done. However, it's also important to remember that quality assurance procedures, as I've said, that quality depends on the perspective you're taking in the application. It's going to vary between research applications, whether these cells are for diagnostics possibly, or for biomedical applications. So the general process that you've already seen described is going from the cell sourcing, the biobanking process, storage, that's producing the cell bank, storing those cells, and shipment, and the input, the raw materials are all important to this. And then checking the traceability and ethical procurement, the freedom to act for any users of the lines, and the quality control. If you get any of these wrong at the start, you could waste a huge amount of money banking cells that are never really going to get out to users. Then also, as we've heard, the testing and quality control that needs to be done on the cell banks, and then the process of release or reject, so a final decision based on all of the information gathered to that point enables you to finally release the cells out. If you don't have a control point there, then you can be sending out cells which are subject to questionable background. And then also, don't forget, as Kevin mentioned, the user acceptance and feedback, and all of these are encompassed under what we call quality assurance. And it's really valuable to have a document, a high-level document, which describes those in outline, the quality manual which we established for EBiSC. So how do we set up the EBiSC quality manual, quality management system and the manual? it needed to cover at least three different centres. So we weren't dealing with just one operation. It had to be something that was flexible to work across multiple centres. So certain aspects of the work had to be devolved to local areas, and we had to decide what was the responsibility at the different levels for the operation. Was it a cross EBiSC protocol that we needed, or could we devolve it down to the local one? And try to keep that as simple as possible, because these things can get incredibly complex very quickly if you try to do everything. So we took an ISO 9000 quality manual template, so that's the quality standard for services, that's a generic industry standard, and that would also enable future accreditation to that standard if it was applicable, but also, in addition to that, would enable translation to some other quality standards which are similar, like the ISO 17025 or the 13485 for medical devices. And then another important element in this was to engage the EBiSC partners, particularly the hot start partners you heard about earlier, in the early stages, in the training workshops, to get their input on the protocols. The protocols were a melded change that we started with a basic set of protocols, which during those discussions they were modified and agreed so that everybody had buy-in to the protocols they were being asked to use. And that, I think, was really important. And then later on, once we'd established the quality manual to go across the sites that were involved, Jane Lukey from Roslin Sells took the quality manual around to the various groups just to make sure that it did fit and it was working for them. In parallel to this, and something which has been quite important because it's actually thrown up some new data on these cells, as Ola just mentioned, is the program of quality control development that we had in place. So we didn't just have a lab which did a standard set of tests that were accepted and go with that. We were trying to improve what was available to people using stem cells. And as you've heard, that was largely work done by Ulrich Shea and Charlotte Chapman, who are here today. Another important element was engaging with the international community, and EBiSC was very active over the years in the International Stem Cell Banking Initiative workshops. This is a group which, if you've not heard of them, bring together more than 20 countries on a fairly regular basis at annual meetings to discuss common issues in the stem cell banking area. And it's not just stem cell banks, regulators come along and expert scientists from something called, a similar organisation called the International Stem Cell Initiative run by Peter Andrews, which has focused on a number of scientific characterization standard approaches in recent years. And finally, I thought, well, I'll take a look at the different areas of application of stem cells and how we might deal with these in terms of quality standards and safety standards, going through three particular phases through professional consensus on best practice. So this is scientific professional consensus. Regulatory guidance developed by regulators and policy makers to help people working in the area to comply with the regulation. And finally, obviously, the law, the regulation as it's implemented from the law. Now, I've looked at here at research, diagnostics, use of cells in diagnostics, in vitro testing, manufacturing of cell-derived products, so things like vaccines and biotherapeutics, and finally the cell therapies where the cells end up as the medicine itself. Not really time to go through all of these legal and regulatory documents, but I thought I'd just pick out on one or two. But the first thing to say is that they're all slightly different. It's not that easy just to translate directly from research to a cell-based therapy. You need to assure certain things around those research cell lines, and it has been done to translate a research cell line to a cell-based therapy, but it's not necessarily the easiest way to do it. A lot of backfill has to go on, a lot of information that has to be dug out to assure the regulator that these cells can be translated to these products. So just to summarize very quickly, one area where there is some commonality between these different applications is something which came out of the toxicology area in the 1990s, 80s and 90s, and that was an agreement on a set of 6 principles of best practice for cell culture. And this works across different cell cultures, types, different applications. So it's a fundamental consensus published by Sandra Kirker et al. In 2005, which is well worth referencing, is still relevant today. Secondly was the interaction that EBiSC has had with the International Stem Cell Bank Initiative, and Kevin mentioned the consensus document on banking human ES cells, which was published in 2010. And even though it's before EBiSC started, members of Rosin Cells and other people here in the room were involved in developing that document. Again, principles of best practice for banking human ES cells. There was also involvement from the ICBI in the WHO document on evaluation of cell substrates for vaccine and biotherapeutics manufacture. That was published in 2010, and there's a section that was drafted by the ICBI group on use of stem cells for manufacture. And then finally, the one that Kevin mentioned, and EBiSC has been active in again, is a consensus on principles of best practice for developing human pluripotent stem cells for clinical application, both IPS and ES. And a number of people in the audience here again were involved directly in the authorship for that paper. So this has been quite an important way of interacting with the community, and hopefully that will continue. The other thing is to mention some new documents on the way through in terms of regulatory guidance, some of which will be effectively, we're very close to the regulation. One of them is an OECD document on good in vitro methods practice, which looks very broadly at development of in vitro method assays and also how to validate them, particularly for toxicology work. And an annex to that will be a revision of the good cell culture practice document, GCCP2, which is under development at the moment. Also, there's an activity called ISO TC150, which is the biotechnology group of the International Standards Organization, producing standards for cell culture work, cell enumeration, etc. And another one, there are developments within the International Conference on Harmonization, so between Japan, Europe, and US, agreed complementary standards on cell therapy development. That's just beginning, I think. And also now something which is now quite advanced, which is the ISO TC276 group, which has 3 themes, one of which is on bioprocessing for cell therapies, and another one on biobanking, which will be important for EBiSC in the future, I think. So just to conclude, I think adopting recognized standards, international standards, has helped EBiSC to be a robust and reputable resource which is relevant to industrial applications. Engagement with key partners in the development in the procedure, so engaging with those hot start labs, the people who are developing the IPS cells, was very important to get it off the ground and shape up the protocols so that they were more broadly acceptable. I think now the EBiSC partnership in its broadest sense, and quite a number of those 26 partners you heard about, are well equipped now to contribute to training in biobanking, and hopefully the EBiSC partnership will live on to do good work in that area. And then there are also new developments. I've mentioned the ISO TC27 group, which is now establishing a biobanking standard, and that's been discussed at the ICBI meetings. The EU directive on data protection, which becomes law in Europe next year in May. So, and we will be affected by that in the UK, even if we're outside of the European Union, because we'll want to exchange data with European researchers. And it will be an issue for anyone working within and between Europe and elsewhere. And that will require rigorous control of patient-sensitive data, and has been in the latest ICBI report for 2016, just been published in Stem Cells and Translational Medicine, and there's discussion on that. And then I think it's also important that EBiSC maintains ongoing engagement with stem cell banking community. The ICBI workshops have been a good forum for that, and hopefully ECAC and others who have already been to some of those meetings will continue that interaction. There's one planned for the ISSCR meeting in 2018. The other thing, I think we've got some good people in the audience here who could talk about the development of clinical grade IPS cell lines, or human ES cell lines in particular, and that's Professor Jack Price, who will be chairing tomorrow, and also Elsa Branches, Al Rocher and Charlotte Chapman are all representing the bank here today. So please talk to them for more hands-on experience in developing clinical grade human pluripotent stem cell lines. I'll just say, I'll give an acknowledgement to Orla and Charlotte, and Mark Toniarelli, who've put a lot of effort in helping to get this program together today on the QA work. Jane Lukey, who had a big role in getting the quality manual taken out to the different centres, and also support from Rachel Steen, Kevin Bruce at Senso, and Beata Kreisel, who's supported us on just about everything we've done in EBiSC throughout the project. And thank you for your attention.
Thank you, Glyn. Questions? There's a question.
So with regards to the regulations, I'm missing the ISSCR guidelines for transferring stem cell research and clinical applications. So you don't consider this to be important? Or, so what's your take on this ISSCR guidelines?
Yeah, I mean, ISSCR's produced a number of guidelines which are helpful, and there are a whole raft of guidelines.
I mean the ones which were released in 2016 now.
So there was there was one which included some elements on banking,
as well as on research with iPS cells and how to deal with the general ethical questions regarding it.
Yeah, I mean, I think those are all helpful, and there has been a whole raft of standards documents, and I've picked on a few that I've been personally involved in and that I can sign up to. So there is a lot of other guidance around. In fact, it's confusing the amount of guidance.
This is the problem with which I see is like.
I mean, I usually try and point, if I'm talking about stem cell banking, I'm pointing to guidance that has involved a large number of stem cell banks from around the world, plus expert stem cell biologists who have been connected with the banks, plus the regulators who are looking at the banks. So if they don't know which way to go, then nobody does. That's my approach to this talk. But there's a lot of guidance on related issues, cryopreservation, storage, other kind of biobanking documents. OECD produced one, ISBA produced one. There's lots of guidance out there. And I'm not saying any of it's bad at all. The ISSCR had been trying to drive this a common ethics consent form, and they've been talking about that for quite a few years. And I really think that's going to be a real challenge. A number of groups have published the components of an ethics document that you might need to have, and I think that's about as far as you can go with the ethics, because every country, every group, every hospital and institution will want to have their control over the ethics documentation, and you will never get a common template form that will work right through all of that. That's my personal opinion.
More question to the talk of Glyn? If not, I would like to thank you again, Glyn.